So, hello everyone. Welcome to this seminar. Today I am happy to introduce Professor Claus Bässler. Claus is a recently elected professor in Goethe University, Frankfurt. So he has been leading his conservation biology group for 2 years. Group has been expanding heavily. And earlier, Claus has been working a long time at Bavarian Forest National Park in Germany. So his research, I think, also carries a lot of experience from that time. And together with his group, Claus is exploring different questions in conservation biology, both more applied and more theoretical, and also covering different groups of organisms. But today he's going to speak about macrofungi. So the field is yours, Claus. Welcome.
Yeah, thank you very much. Can you hear me? Yeah. So, thanks for this nice introduction, Katri. I'm quite happy to be here. Thanks for the invitation. It's an honor to be here. Yeah, because mycology and fungal ecology has a long tradition here. Also with, for example, Leo Tattersall having a lot of really groundbreaking papers in this field. So I learned a lot from Leo. And yeah, I came very late to science. I finished my PhD in 2008, and I thought I have to learn more about fungi. So I joined in 2009 the Fungal Ecology conference in Edinburgh. And at that time, Leo had his first keynote, and he was very young. And I thought, oh, he is a really nice guy and makes really interesting things. I would also like to do such things. So this is the connection what we have. And so I'm happy to be here. As Katri mentioned, the title of my talk today is Macro Fungal Functional Ecology Towards a Deeper Understanding of Fungal Diversity. It's a bit ambitious, I agree, but it is what is my main focus and my main interest, to better understand diversity pattern. And probably I speak out of your heart that long time fungi were the forgotten kingdom in ecology, and particularly also macroecology. And there might be 3 main reasons. One reason might be that long time distribution data at sufficient grain size and extent were missing compared to the other kingdoms, for instance, the plant or animal kingdom. But in recent years, we made important progress also because of this group here in gathering data across the world to address pressing macroecological questions. From these studies, we learned that particularly climate, but also variables related to resource availability, which is very important for fungi, play a pivotal role in driving the diversity pattern at larger scale. And second, if we would like to gather a deeper understanding of the diversity pattern on the assembly processes, we need information of assembly relevant traits across thousands of species. And this is also not easy to reach, also not easy from meta-barcoding data. We also need to address these kind of questions, megaphylogenies, but also here in recent years, we made important progress to push fungal ecology forward. And the third point is, and this is a bit of my narrow view, and Katri also introduced it, I think also if we would like to understand better the diversity pattern, we have to set up experiments to deal with the many confounding effects which exist when having only observational data. I'm pretty aware that it is not easy to reach at larger scale, but yeah, we have to start at some point. And also forest ecosystems are a nice model system in that respect, because variables related to resource availability like deadwood and abiotic factors like microclimate caused by canopy change are normally heavily confounded in these systems. In my research, I try to cover a broad range of concepts and recently also methods. So I'm interested in within and across species variability, but particularly how differences among species translate into assemblages. And here I also try to consider different spatial scales. I'm using observational data in my studies, but also experimental data, and I'm interested in different kind of methods. My history is fruit body inventories, but this is only half of the medal. We are pretty aware today about the need of using different methodological approaches to have a better understanding of what is a fungal community. I'm using various skills or lifestyles, but I'm particularly interested in saprotrophic fungi. And the environmental context I'm working in is climate, macroclimate and microclimate, depends on scale, and resource availability. Both factors translate into what we call anthropogenic change, climate change, but also forest management intensity, which is mainly affecting resource availability and resource diversity, affecting fungal diversity in forests. I would like to give you today a few examples about this framework, and I'm starting with a very coarse scale. From Leo and co-workers, we learned, and also from a few studies at continental scale where I was involved, that climate is an important factor driving fungal species diversity. Temperature plays a major role among climate variables, also precipitation. But the mechanisms are poorly understood. From the animal kingdom, we have evidence that variables related, or the mechanisms, thermoregulations play a major role in the distribution of species. You might know Bergmann's rule, for instance. So thermoregulation hypotheses have been developed for more than 100 years in other kingdoms, but rarely adopted and applied in fungi. Many species rely on sexual reproduction and to maintain populations. And the sexual reproduction origin of many fungi, particularly macrofungi, are the fruit bodies. The fruit bodies are exposed to the environment and should therefore be, or the prevalence of fruiting species should therefore be correlated to the thermal environment. So we tried to develop a thermoregulation hypothesis and thought that two traits of fruit bodies might be particularly important, also from prior knowledge and from other ectotherm organisms. The one is the size of the fruit body, and the other trait is the color. Hypothesis related to the size is not straightforward. There are two opposing hypotheses. The conservation hypothesis would state that on average, the fruit body size, or the fruit body size of assemblages, is hump-shaped related with temperature, which buffers large fruit bodies, buffers to extreme temperatures at the warm and at the cold edge. The heat up, cool down hypothesis, by contrast, would predict that fruit body size on average is unimodal or hump-shaped related. They are smaller at the edges to get rid more rapidly from heat or cold stress. We have no idea about that. The thermal melanism hypothesis is more straightforward. It has been developed from the insect kingdom by Ernest Rutherford. And here the prediction would be that fruit body lightness of assemblages would increase with temperature. So on average, fruit bodies should be darker in cold environments to be better able to harvest thermal energy. This would be the hypothesis. To address the first hypothesis, we gathered a data set of fruit bodies and fruit body size across the world. And what we found is that the assemblage fruit body size shows a unimodal relationship with temperature. This would support the heat up, cool down hypothesis. We also found the same relationship at species level. So at least fruit body size contributes to some extent to global assembly of fruit body forming macrofungi. To address the thermomelanism hypothesis, we used a data set which has been gathered within a larger consortium. It consists of citizen science data and also herbarium records. And we color-coded, my former PhD student Franz Grah, more than 3,000 species, which you can see here, and to develop a lightness gradient of the fruit body colors. And here are the results. As you can see here, the assemblage lightness correlates positively with mean annual temperature. These are partial effects from more complex models where we also considered UV or precipitation as a covariate. And yes, you see a lot of scatter, and it also holds true not only for saprotrophic fungi, but also for ectomycorrhizal fungi, which points to at least, some, I wouldn't say evidence, because there are also other aspects important in that respect, but at least we found a positive correlation which supports this thermal melanism hypothesis. So both size and fruit body color might be important for the assembly of macrofungi at larger spatial scale. But using only the one or other trait is surely not enough, and considering phylogenetic information among species might help to get a better understanding. And the basic idea is that species adapted to specific climate conditions are located within lineages, so some kind of trait conservatism. So we used again this pan-European data set, and we expected that we will find a phylogenetic clustering, that species are phylogenetically more similar than expected within grids or plots at the end of at the edges of the temperature gradient. We not only focused on mean extremes, but also on temperature seasonality, because a high variability and an occurrence of extreme temperatures might also filter towards species from specific lineages if this trait and adaptation is evolved in a conservative way. This is more or less on phylogenetic alpha diversity. We also considered phylogenetic beta diversity. So in simple words, we tried to address whether species that are subject to different temperature regimes or environments belong to different or disparate lineages. And if this would hold true, like indicated here, we would expect an increase of phylogenetic beta diversity or a dissimilarity of phylogenetic assemblages with temperature distance or similarity. What we found is on alpha level that indeed the relationship is hump-shaped, which supports this habitat filtering hypothesis. So phylogenetic diversity is more similar than expected at the temperature edges. But we found, by contrast, not a negative relationship that strong seasonality filters to specific lineages, rather than it shows an over-dispersion where the species are phylogenetically more dissimilar than you would expect by chance. Probably one explanation for this is that the temperature niches are more complex in temperature seasonal environments, which explains that different species from different lineages occur in such kind of environments. When focusing on beta diversity, we found a positive but a rather weak relationship with mean temperature distance and almost no relationship with temperature seasonal distance. This means that the species-rich genera occur in different environmental conditions. And this effect, this small effect, is caused by a few species-poor genera which exclusively occur in extreme cold or warm environments. So the large lineages occur across the temperature range, but a few genera like Amylocystis, for instance, you know that, and others occur exclusively in cold or others in warm environments. Whether this is also influenced by biases due to because these are citizen science data, I cannot answer this sufficiently, but at least this is what we found. So macrofungal assemblages are phylogenetically structured by temperature. All these measures are based on null models, so at least it deviates from null expectations from random. So at least we can say that macrofungal assemblages are structured by phylogenetically structured by temperature. And phylogenetically constrained specialization, particularly at species level, might exist towards temperature extremes. This is what this study is telling us. Coming back to this thermal melanism aspects, the temporal resolution of the data set was rather weak, but at least covers 40 years. And if the color of assemblages is mechanistically linked to temperature, we would expect that with temperature change due to climate change, and change of the assemblages based on their colors would be observable. So we simply plotted the change in color lightness of assemblages with temperature change during the 40 years and found no change. It might be that the data set is too weak, or that changes in thermal respect is based only on abundances or abundance changes. At least there is not a strong turnover. And this points to the fact that we have to be careful about such kind of studies, and that phenotypic variability or also physiological adaptations might species to enable to cope also with temperature changes. And yeah, we run after this in the lab, and it is just a small and rough study, but address the question whether this model species, the fruit body, if we expose it to temperature extremes at 5, 30, and 60 minutes, the fruit body is still attached to the substrate, not to confound the results, to check whether there is some kind of physiological adaptation. And we run some transcriptomics and found indeed no change in morphological structure after 60 minutes, and the expression of a lot of proteins which points towards coping with heat stress. Also, different co-class enrichments were observable, which points definitely towards defense mechanisms. It's a first approach, but we were inspired by particularly this results, and we reconsidered what we did so far, and wrote an opinion paper that it's necessary to consider much more intraspecific trait variability, phenotypic variability, because if you only have a distribution data set and you assign the traits based on literature, this might be a pitfall, or there might be some pitfalls, because then if you detect a change, is a change in turnover, but there is happening something in between. So we would need to measure, and you can imagine that this is all but easy, to measure for thousands of species the community trait mean based on specific site-dependent trait values. So you have to go out in the field and then not only count mushrooms and assign them a name, but also to measure the traits in situ. Because if this trait is mechanistically linked, I would even expect a stronger response. This would be the prediction. But it might also be that there are spurious correlations, which we observe today. And from the plant kingdom, we learned that even the assembly process can be wrong when only using community fixed means based on literature compared to if you make really in situ measurements. So we should be aware about that. But this is only a side aspect. My next example, I would like to show you a few results from a cross-species approaches. And to have a short introduction, above all, fungi need resources, carbon and nutrients for growth and reproduction. And there are two important aspects of resource availability. The one is the lifestyle, how the species is gathering the resources needed for growth and reproduction. And the second aspect is the resource availability provided by a habitat, for instance, measured in carbon stock or netto primary productivity. You are excellent mycologists, so you are pretty aware that there are two basic lifestyles. The saprotrophic fungi have to produce rather costly enzymes to break down organic matter, while ectomycorrhizal mutualistic fungi gain carbon from their host in exchange for water and nutrition. According to mutualism theory, a mutualistic relationship should increase the fitness. And this is a nice model system where we can use the saprotrophs as a reference group and test mutualism theory. Mutualism theory has rarely been tested in fungi. And we used the fruit body size and the reproductive biomass of the fungi as fitness measures. So in a first step, we asked if this holds true, ectomycorrhizal fungi should on average, on species level, larger than saprotrophic fungi. And we applied a phylogenetic informed analysis about 600-700 species, and indeed found, this is these are the effect sizes from this phylogenetic model, that on average, the fruit bodies of ectomycorrhizal fungi are to 1/3 larger than those of saprotrophic fungi. And interestingly, when using field observation data, the fruit body size, fruit body number trade-off, because there should be a trade-off, because also for mutualistic fungi, carbon is to some extent limited. So you can either invest in a few large or in many small fruit bodies. But interestingly, this size number trade-off is much less pronounced in mutualistic fungi compared to saprotrophic fungi. The fact that they are on average larger and have on average more individuals, or the trade-off is less pronounced, leads then indeed to a higher level of reproductive biomass compared to saprotrophic fungi, with consequences on population dynamics, radiation, whatever. The next consequence steps would be to test whether this translates into assemblages, and how, if resource availability is related to fruit body size of assemblages, how this is correlated then with resource availability of the habitat. This is what I already explained. So we developed two hypotheses, resource availability hypothesis. The one is a very simple, the ecosystem's biomass hypothesis, which would state that fruit body size should increase with the biomass of the habitat. And a second hypothesis, we were inspired from the animal kingdom, is the seasonal resource surplus hypothesis, because seasonality is regulating the quality of resources. And Hasten and Wolferton suggested a measure called ecologically evolutionary netto primary production, because they showed that the resource availability during growth and reproduction is correlated with the time of the availability of resources. So fruit body size should therefore also increase with seasonality, with climate seasonality, or this ecological evolutionary netto primary productivity. To test the first hypothesis, we used a local data set characterized by huge differences in growing stock among forests, and found for both the ectomycorrhizal fungi and the saprotrophic fungi an increase of mean fruit body size of assemblages with resource availability. The strength of the response is more pronounced for saprotrophic fungi compared to ectomycorrhizal fungi. And when plotting the mean fruit body number with resource availability, you see a negative correlation, which again indicates this trade-off. And you also see here that the relationship or the strength of response is more pronounced for saprotrophic fungi compared to ectomycorrhizal fungi, which also supports mutualism theory. At global scale, we do not find this pattern. These are the partial effects from this model I showed in previous slides, where we plotted the mean fruit body size against temperature. But at global scale, we found a strong relationship of fruit body size with this seasonality, or ENPP, which supports the seasonal resource surplus hypothesis. So also resource-related factors might affect fungal assemblages via fruit body size, also at different spatial scales. Now I come to my last example. Third, I claimed that we need a bit more experiments at different spatial scales to disentangle confounding effects. And when you look at this forest and you're interested in deadwood organisms, you wouldn't be surprised to find a lot of species there, simply because the number of species is related to deadwood amount. This is quite trivial. The problem is, however, that the diversity of deadwood is also correlated with deadwood amount. You have not only a lot of deadwood, you have also a lot of different deadwood. And the other aspect is that if you have a disturbance event, or you have some such kind of these conditions, the resource-related variables are also correlated to the environment, for instance, microclimate. So all these factors are confounded. And if I would like to make concrete recommendations for to a forester or for forest management, it is helpful to know exactly the effect, because if it's all an effect of resource amount, I have to say to the forester, sorry, you have to leave a lot of deadwood, otherwise we lose species. But if it's deadwood diversity and combinations, a clever combination of different kinds of deadwood resemble also a high number of species, you can also use wood. So this is my second interest in my research. So we carried out, I would not like to go into detail, a few experiments on different spatial scales within Germany, from object scale to stand scale. And recently we also set up a larger global deadwood experiment, but it's much more easy and not that complex. And we did also not manipulate forest stands. We used in these experiments different kind of methods, still fruiting body inventories, but also meta barcoding. And recently we also started to capture the airborne species pool, but have not so much data yet for this kind of community. I would like to show you two examples from these experiments. In a very, very first study, we were interested whether the diversity of one specific object is affected by the immediate surrounding of deadwood, when you have a high level of deadwood or a low level of deadwood, or a high level of diversity of deadwood and a low level of diversity of deadwood. The rationale is that I would expect that diversity might boost if you have a large donor pool close by to have more species on an object if you have a lot of diverse deadwood around, rather than if the log is surrounded by less amount or diversity of deadwood. We were interested whether the microclimate plays an important role, and of course we were interested whether the identity of the tree, whether I'm a fir or a beech, and I have fine woody debris or coarse woody debris. So we also manipulated the log itself. And when focusing on these results, you see that factors related to the host identity, mainly tree species, plays a major role in driving the number of species. The environment, whether there is a lot of deadwood, there is no deadwood, whether it's open or it's closed, seems to play a rather minor role for the number of species. The environment plays an important role for the community composition, but the main factor still is also here the host-related factors, and this is, am I a beech or a fir? But by the color you might recognize, and this is the second axis, which is showing this, is a change also of the fruiting community with microclimate conditions. We were inspired by these results and asked what specific characteristics have these fruit bodies, and can we explain this shift based on fruit body traits? You are now aware about the role of size and color for thermoregulation. So thermoregulation might play a role in this microclimate context, but also variables related or traits related to the balance of water, transpiration or water holding capacity. If you lay a agaric in the sun and a polypore, it's quite simple what is happening. So we derived these three kind of characteristics. We tested whether the change in microclimate conditions is related to the size of the fruit body communities, to the color of the fruit body communities, and we coded a toughness measure to have a measure of water holding capacity, according to the literature. And yeah, here would be the hypotheses, which are also not straightforward, so I would not go too much into detail. I tell you immediately the results. The only measure which were consistent across our analysis was indeed the toughness. And fruit bodies are on average tougher in open conditions compared to closed conditions. And this holds true for different kind of fungal guilds, and also is consistent across tree species, which points to the importance of this factor. And in my last example, I would like to show you results from my current PhD student. Conservation is always a matter of scale. This is what you're pretty aware. And we have an experiment running in Germany across larger scale, where we have plots located across landscapes, which reflect differences in macroclimate conditions, but also in forest communities. We have plots within these regions which reflect differences in stand scale characteristics. And on each plot, we have 13 different tree species from these two main lineages, from the gymnosperm and angiosperm lineages. And we simply asked, what is more important? Is the space more important? Is the host more important? Within the space, is the large scale more important, or is the small scale more important? And at the host scale, we ask whether it's more important to have conifer versus broadleaf tree species, or within these lineages, different kind of tree species. We tested this relative based on suboxylic beetles, fruiting communities, molecular derived communities, some kind of mycelial communities, not sure this is the appropriate term, and bacteria. Yeah, and it's a bit annoying because when focusing on beetles and the fruiting community, all of these factors seem to some extent important. So there's no simple strategy that we can say, oh yeah, you do not need to consider the spatial arrangement of protected areas, because if the host scale matters, it doesn't matter where you locate a protected area, for instance. So we tried also to assign specific conservation strategies if some of these factors would be prevalent. But you see that this is not the case for subroxylic insects and the fruiting community. All of these aspects play an important role. So you have to consider deadwood of various kinds from different lineages across different regions, so to say. But I was struck by these results when focusing on the molecular community. You see that the host is much more important than the space. And this is a fungal measure and this is a fungal measure. And you would come to totally different conclusions when having only this or having only this. I would like to make one example. So basically it seems the fruiting community is more sensitive to the environment than the communities derived within deadwood from molecular metabarcoding. If I would have only this result, I would say the spatial arrangement of protected areas makes no sense. Make protected areas where you have a lot of different tree species. But then you would ignore the areas in which at least the macrofungi can sexually reproduce, build spores and maintain populations. We have not that very pronounced knowledge about dispersal capability in fungi, but if they are dispersal limited and they cannot reach the habitat to maintain the populations, then the populations in the specific regions would go deplete. So it was for me very important to consider different methods if it comes also to such kind of conclusions. And of course, we need different deadwood tree species on a broad spatial coverage at the national scale to maintain diversities. This is unfortunately not a simple recommendation. Okay, then I come slowly to the end, and I would like to tell you what I'm interested in and would further try to boost. I would like to learn more about intraspecific mechanisms to cope with climate change. And I also would like to consider not only the sporocarp or the spores, but also the complete life cycle. So we have an application running at the moment, which is under review, where we consider species-specific aspects, but also community-specific aspects. Then we're analyzing at the moment this global experiment to address the question about the latitudinal diversity gradient of wood-inhabiting fungi. We have only knowledge from soils so far. And if it holds true that the tropics also harbor the most species compared to higher latitudes, we would also like to approach the mechanism behind. And of course, wood-inhabiting fungi are specialized organisms which depend on the host. And therefore, one hypothesis would be that if it holds true that the tropics harbor the most species, this might be caused by niche partitioning or niche packing, which is illustrated here, that on average, a deadwood object harbors more species in tropical regions compared to higher latitudes. And I'm quite happy that I was able to hire Katri for a specific smaller project where we are particularly interested to learn more about the relationship between fungal diversity and the ecosystem processes which are related to this diversity, and particularly considering a broad range of lifestyles. And during our first work, during Katri's first work, it turns out that a lot of aspects are missing at the moment. So this is what we are also working on. Finally, I would like to thank my team and a lot of nice collaborators. And I thank you for your attention. Of course, I'm happy to take questions.
Thank you, Klaus. Impressive talk. Questions, anyone? Well, Professor Urmas Kõljalg, please.
Thank you for the talk. I'm interested about the first part, of course, this database of traits from sporocarps. It's so, but anyway, some results you showed us were for species diversity, alpha and beta diversity. So I'm a taxonomist, so my interest is to hear how it is possible to have sporocarp data and then to analyze species and then even alpha and beta diversity, because we are talking here data about sporocarp formation.
Yes, we are talking
very little, almost nothing about the individuals as such, and then even higher level species. Am I correct?
So the database, I have a name to a fruit body which has been observed in a specific environment and in a specific location. And these analyses are simply then based by gridding this data and having, for instance, Rusula ochroloica, Amanita muscaria, Polyporus squamosus, whatever on this grid. So these analyses are based on such kind of data. And of course, citizen science data have their drawbacks. Are also presence only data. So we did a lot of effort to try to make the communities robust. For instance, considering a minimum species per grid for the analysis. If there is only a few records in a grid, we removed it simply. So here at least 50 to 100 species are in a grid, which we consider then for the analysis to have some kind of robust community. Carrie Andrew, in her first paper in this Journal of Biogeography, was the first which came out after compiling this data set, did a lot of such kind of sensitivity analysis. So, and I relied on these first papers and applied the similar thresholds, produced this mega phylogeny, and then focused also on beta diversity or phylogenetic beta diversity.
Oh, right. It was phylogenetic, not ecological.
This was phylogenetic beta diversity. No problem.
Thank you for a very interesting talk. My name is Kadri Põldmaa and I work as a mycologist here. It was very nice to see you comparing fruit body and molecular method-based data in connection to nature conservation. First, do you know any cases in Germany or elsewhere where already these molecular method-based data would be taken into account in making nature protection decisions? Or second, what is your opinion how we would now start incorporating that enormous knowledge we have already based on sequence data in contrast to this decades-old tradition of using the fruit body data for all sorts of decisions? Thank you.
Yeah, it's not really developed in Germany, as you may expect. So the red lists and many conservation actions are still based on these fruit body inventories. So these meta barcoding data at the moment are not heavily used for practical conservation or recommendations, but this is developing. I think it's important, even though both methods have their advantages and disadvantages. That's quite clear. Of course, if I have a fruit body, I do not know if the fruit body is not there, whether the species still exists as mycelium. I would expect this. So we are trying also to approach under which conditions a species is prevalent as mycelium, but do not produce fruit bodies because of environmental constraints. So we have to learn much more about the prevalence of both aspects of these organisms. And yeah, and of course, meta barcoding data have also their advantages and disadvantages. The species assigned, the name must not necessarily be true. It's also as good as the database is, but this is also super developing. Omas and colleagues work heavily on such kind of things. So I think we should think in future that, go forward and consider both aspects. I would be rather interested to compare both fruit body inventories and meta barcoding data also at larger spatial scale. But it's really a challenge because the grain size of meta barcoding, it's a piece of soil, and of course these fruit body data integrate across 10 by 10 kilometer the data we have. So it's not easy to combine them. So we would need an appropriate grain size and extent, having both data to learn more about prevalence of mycelia and the production of sporocarps. And also then to make concrete conservation, of course. I'm not sure how it is in Estland, in Estonia. Mm-hmm.
I think we just finished more or less completed red list, the new version. And I think most mycologists, they were using eDNA data, and then the results are extremely interesting because you will find very rare species described in Finnish taiga region, which are now present based on eDNA, similar forests, no sporocarps found. So all kind of very interesting findings.
Oh, I can imagine. Yes. It's only a part of fungi produce these conspicuous fruit bodies. It's not the majority necessarily. So I totally agree that it is. Probably you can tell me more about that.
Hey, Claus, nice to meet you. Mohammed Bahram, associate professor here. As an ecologist, I was really interested in your hypothesis about latitudinal diversity gradient. But I wonder if, I don't know if there is any study looking at wood decomposition in tropics and versus temperate, and whether, which I could imagine woods would be much quicker, much more quickly decomposed in tropics, and whether that would affect the diversity of wood decaying fungi on wood. I mean, considering if you give them more time, that they tend to accumulate more fungi, and whether that would not interfere with this latitudinal diversity gradient.
Mm-hmm, mm-hmm. So you mean that if the turnover or the decomposition rate in tropical forests is much faster, then of course the development of the diversity of the species is different. Yeah, that is definitely the case. We try to approach this also based on this data set. And the data set we are focusing at the moment on is to standardize for the early successional stage to make it indeed comparable.
Okay.
Yeah, because otherwise you would run also in these kind of pitfalls you mentioned. Yeah, sure. Definitely.
Yeah, thanks.
Thank you for a very inspiring talk. I'm trying to formulate the question, but it may be like that. What, in your opinion, is actually the appropriate variable, climate variable, when we analyze the diversity of fungi? So I try to explain it that you have fruit body data, which in many cases are very short-lived. And then you have climate which by definition is a long-term phenomenon. And then among the fungi, we have individuals which may be of some intermediate lifespan, and then we have species or lineages which are very long. And on this gradient, I somehow see a mismatch that we are comparing short-lived fruit bodies with long-term climates. And maybe this is one of the reasons why we see these very weak and fuzzy relationships. But instead, maybe we should be comparing fruit bodies with some short-term climatic events and then some larger or long-term units of fungi with some longer-term variables. Is that a question?
No, but you also give the answer. This is an excellent question. You're totally right. Of course, a fruit body or a species might adjust to avoid unfavorable windows of opportunity, or use the optimal window of opportunity. But you shouldn't forget that also fungi are constrained by other factors. This might be intrinsic on the genetic basis, or whether a host provides resources to a specific time. But anyway, I don't, you're totally right. The weak relationship and also the scatter might be because there are many more strategies of fungi. Here we are only considering whether on average cold temperatures, is the species prevalent and is it able to produce fruit body or not? So this is very coarse, but I'm quite sure, and you have definitely right, that we have to consider the seasonality and the relation to seasonal temperature or precipitation, because this is happening on the individual scale.
So maybe the follow-up question, then the real question is, what kind of climatic variables you have tried? Because in vegetation ecology or plant ecology, they use sometimes very specific, like the number of days below certain temperature or so on. So what's your approach?
Yeah, a rough approach, because simply at that scale, many temperature variables are correlated. And then we simply use mean temperature as a proxy, because the temperature of the growing season, for instance, is closely correlated to the mean annual temperature. And so, yeah, it's a quite simple approach. And of course, I can run thousands of models with 19 bioclimatic variables. But then finally, I did this indeed. And finally, I stick to mean annual temperature because it's some kind of integral across, which was the most suitable. But of course, I'm not sure whether this is the causal factor in such kind of analysis.
So I'm next. Hi, thank you for the nice talk. I'm Nilufar, a postdoc, also ecologist. So my question is more about the resource availability theory that or hypothesis that you had. So I was very surprised that the graph that you showed was totally linear. I was expecting that it would be flat at some point, like the fruit body size doesn't respond to the resource availability at some point, and then the line would become flat at some point, even if we don't consider competition or like increasing the biodiversity and so on. I would expect it to be flat at some point. If you explain a bit.
This is definitely also a question of the scale and whether you reach some upper limit. And the results were shown on log scale. So this makes also a difference. But the gradient we used here, for instance, this local gradient is not the total range in which the fruit body size is occurring. So it's a narrow window, so to say. And there is some saturation, but we use the log scale on the y-axis.
Hey, video.
Hi, very interesting presentation. And I would like to ask you about this relationship between fruit body color and temperature. was based on latitudinal gradient or latitudinal and elevational gradient?
Both are inside this data set because these large parts of Europe have this latitudinal temperature gradient, but also parts were from the Alps, for instance.
Mm-hmm.
And we simply regressed it for this reason against mean annual temperature of a specific grid to make one measure out of latitude and elevation. But we did in the supplement a separate analysis where we showed the same relationship with altitude.
Okay. Did you observe also differences between the different groups of fungi?
No, not between saprotrophic and ectomycorrhizal
Okay.
fungi, which makes hope that there's at least some relationship.
Okay.
Yeah.
Okay.
Even though the ectomycorrhizal fungi are on average darker, which also points towards mutualism providing advantages for the species because pigments are always also costly.
Mm-hmm.
It's also a side aspect of this study.
And my last question is, do you think that this relationship could be also extended to other regions?
I would expect that we will find, on average, darker fluid body assemblages in cold environments.
Okay, thank you.
If this is a mechanistic filter, this would be the prediction, but we should test it in other regions.
In absence of other urgent questions, I think we should start thinking about finishing because 10 minutes over time. But I would like to ask the last question or make the last comment. You showed many world maps, but never had you a dot on Estonia. So
This is the reason I'm here.
so hopefully that will change then. But thank you very much, Claus, and let's
Thank you.